Assalamu alaikum. Today topic is epitope mapping. First of all, like and subscribe my YouTube channel. Then we start our topic. Epitope mapping. T cell epitopes. MHC class 1 and 2 epitopes can be reliably predicted by computational means alone. Although not all in silico T cell epitope prediction algorithms are equivalent in their accuracy. There are two main methods of predicting peptide MHC binding: data driven and structure based. Structure based methods model the peptide MHC structure and require great computational power. Data driven methods have higher predictive performance than the structure based method. Data driven methods predict peptides MHC binding based on peptide sequences that bind MHC molecules. By identifying T cell epitopes, scientists can track phenotype and stimulate T cells. B cell epitopes. There are two main methods of epitope mapping, either structural or functional studies. Methods for structurally mapping epitopes include X-ray crystallography, nuclear magnetic resonance, and electron microscopy. X-ray crystallography of antigen antibody complexes is considered an accurate way to structurally map epitopes. Nuclear magnetic resonance can be used to map epitopes by using data about the antigen antibody complex. This method does not require crystal formation but can only work on small peptides and proteins. Electron microscopy is a low resolution method that can localize epitopes on larger antigens like virus particles. Method for functionally mapping epitopes often use binding assays such as Western blot, dot blot, or ELISA to determine antibody binding. Competition methods look to determine if two monoclonal antibodies can bind to an antigen at the same time or compete with each other to bind at the same site. Another technique involves high throughput mutagenesis, an epitope mapping strategy developed to improve rapid mapping of conformational epitopes on structurally complex proteins. Mutagenesis causes randomly site-directed mutations at individual residues to map epitopes. B-cell epitope mapping can be used for the development of antibody therapeutics, peptide-based vaccines, and immunodiagnostic tools. Next, epitope tags. Epitopes are often used in proteomics and the study of other gene products. Using recombinant DNA technologies, genetic sequences coding for epitopes that are recognized by common antibodies can be fused to the gene. Following synthesis, the resulting epitope tag allows the antibody to find the protein or other gene product, enabling lab techniques for localization, purification, and further molecular characterization. Common epitopes used for this purpose are MIC tag, HA tag, flag tag, GST tag, 6X haze, V5 tag, and OLAs. Peptides can also be bound by proteins that form covalent bonds to the peptide, allowing, allowing irreversible immobilization. These strategies have also been successfully applied to the development of epitope focused vaccine design. Next, epitope based vaccines. The first epitope based vaccine was developed in 1985 by the Jacob et al. Epitope based vaccines stimulate humoral and cellular immune responses using isolated B cell or T cell epitopes. These vaccines can use multiple epitopes to increase their efficiency. To find epitopes to use for the vaccine, in silico mapping is often used. Once candidate epitopes are found, they construct are engineered and tested for vaccine efficiency. While epitope based vaccines are generally safe, one possible side effect is cytokine storms. So it was all about epitope mapping and vaccines.